This video will discuss two methods of humanizing animals, either by introducing human tissue to the brain of the animal or by engineering animals with human genes. Let's start with the former. A non-human animal with populations of human-derived cells is known as a human-animal chimera. Fans of the show will remember our discussion of human-pig chimeras. In genetics, an interspecies chimera is a living thing with cells from two different species. In science, like in mythology, a chimera is an amalgamation of two or more animals. A human-pig chimera, then, is a living thing with cell tissue from a man and a pig. Research led by Dr. Jun Wu and Dr. Juan Carlos Espucia Belmonte at the Salk Institute in La Jolla, California, introduced human cells to the early embryo of a pig. A small portion of these human cells were able to survive in the pig embryo as it grew in a dish, outside of a womb, and the experiment was terminated before developing too far. These interspecies creatures were never carried to term. They never took a breath. Here we see the pig embryo with the human cells labeled in green. The human cells introduced to the pig were stem cells, cells that had the potential to specialize into any cell type. After fertilization of the egg by a sperm, during early development, the cells making up the embryo are stem cells. These stem cells can become any organ: skin cells, blood cells, or even brain cells. So why would anyone want to put these cells into a pig? Scientists wanted to create pigs that can grow human organs for transplants. If they turn off the genes responsible for making the heart, the pig will die. If they introduce human stem cells, human cells with the heart gene turned on, the human cells will fill the niche, specializing into heart cells. Then, the heart, which is made out of human tissue now, can be removed from the pig and transplanted into a human. But why not just use a normal pig heart? Well, since the chimera's heart is made up of human cells, the immune system, eager to attack any foreign material, will be less likely to reject the transplant. If the heart was made of normal pig cells, a healthy human immune system would destroy it, rendering the transplant a failure. Thousands of Americans die every year waiting for an organ transplant. There aren't enough organs to go around, so some are trying to make humanized animal organs available. Ostensibly, this is a noble cause, but the research is far from uncontroversial. In 2005, the year preceding Tim Allen's impressive performance in The Shaggy Dog, Kansas Senator Sam Brownback proposed legislation to make the creation of a human chimera a felony. This act sought to ban, quote, a non-human life form engineered such that it contains a human brain or a brain derived wholly or predominantly from human neural tissues. President George W. Bush learned about this proposed legislation and urged Congress to, quote, prohibit the most egregious abuses of medical research, which include creating human-animal hybrids. The bill never passed, and opponents of chimera research never got to say, mission accomplished. In 2021, the team who brought us the human-pig chimera have created a new human-monkey chimera. Funded by Chinese institutions and private foundations, Belmonte, Wu, and their collaborators in China implanted human stem cells into macaque monkey embryos and grew them in a petri dish. The embryos were kept alive for 19 days, which is the maximum amount of time medical science can keep these little squirts alive outside of a uterus. At that point, the embryos were still microscopic clumps of human and monkey stem cells. Here, we see the young monkey embryo with human cells labeled in red. Bioethicist Dr. Insoo Hyun makes the point that there are no official plans to implant the human-monkey chimeras into the wombs of mother monkeys to carry them to term. If such a chimera were viable, the humanized organs would be too small for human implantation. Livestock donors, like pigs and sheep, are being considered as potential viable walking, breathing human-animal chimeras because the size of their organs are compatible with that of humans, and their lifespan and the existing infrastructure surrounding animal husbandry makes it ideal for widespread organ harvesting. Dr. Belmonte's human-monkey embryos are primarily used to better test human stem cells to make them more compatible with these other species, and this is easier to optimize in our close primate cousins than in more distantly related animals like pigs. The human cells have an easier time thriving in a monkey than in a pig, so this research helps scientists tinker with the cells so they can one day better take hold in a livestock animal. In the US, the National Institutes of Health have issued a moratorium, a halt on funding research that involves putting human cells into the very early embryos of non-human animals. This research is still perfectly legal, it's just that Uncle Sam isn't going to fork over taxpayer dollars to let us do it, though this could change very soon. The main question on everyone's mind is, what is it like to be a human-animal chimera? Can human cells grow in the brain of a monkey? And how human would that brain be? Now, a stem cell only differentiates into a specific cell type, such as a brain cell, if it receives the proper chemical signals from its surroundings. These signals turn genes on and off. These genes are responsible for the identity of the cell. In a typical human, almost every cell in the body has the same genome, the same genes. So why is it that brain cells and muscle cells are so different if they both have the same DNA? Cells specialize based on what genes they have turned on. Brain cells have brain genes turned on. Muscle cells have muscle genes turned on. In the brain, muscle genes are turned off, and vice versa. As an embryo, you start out as a ball of stem cells. As the embryo develops into a fetus and then a baby, these cells start to specialize, becoming specific organs and turning on specific genes. Stem cells become specific cells depending on the chemical signals they are bombarded with in that region of the body. Signals in the head tell stem cells to turn on brain genes, while signals in the bicep tell them to turn on muscle genes. So what happens to a human stem cell inside of a monkey embryo? The human cells start to respond to signals from the monkey cells. An analysis of genes turned on in the human cells of the chimera revealed that the chimeric human cells resembled monkey cells more so than typical human cells. The signals from the monkey cells made the developing human cells more monkey-like. At a molecular level, the monkey cells influenced the human cells and vice versa. If the embryo could grow to the point where it started to develop a brain, and if human cells had made their way into that brain, they would become somewhat similar to the monkey brain that surrounds them. So, here we have monkey embryos in a petri dish lightly seasoned with human cells. The human cells have been somewhat influenced by the simian milieu surrounding them and are starting to act like monkeys themselves. Despite this, many are skeptical that at this point a human monkey embryo would be healthy enough to carry to term. While they found that all embryos did have surviving human cells a day after injection, about half of the chimeras stopped growing after 9 days. And some chimera researchers, like Stanford's Hiromitsu Nakauchi, are looking into making stem cells that cannot become brain cells, to avoid any semblance of a human soul trapped in their chimeric creations. But there are scientists out there who have intentionally put human brain cells into animal brains. But first, what are brain cells? How do these pathetic sacs of protoplasm give rise to intelligence, consciousness, and human emotion? Well, the two broad classes of cells in the brain are neurons and glia. Neurons are the cells we traditionally think of as brain cells. Neuroscience is the study of the brain, and neurology is the field of medicine dealing with the brain and nerves. Glia are the brain cells that, among other functions, support the neurons and help them thrive. There are many types of glia, each with specialized tasks. Neurons act like wires that network with other cells, and like wires, they fire electric currents. The connections between neurons are the basis of thought, learning, intelligence, and behavior in anything with a brain. How would these wires facilitate learning? Well, it turns out that new memories are made when new connections form between neurons, and that connection gets stronger the more times it is activated. Whenever a neuron fires an electric signal, it releases potassium and chemical signals that activate or inhibit the connected neuron. If too much potassium signal lingers in the space surrounding the neuron, it will fire less often. Potassium is positively charged, and like the positively charged ends of a magnet, potassium repels potassium, like repels like. To fire again, the neuron must release potassium, but the positive molecules will not leave the cell if there is even more potassium just outside, blocking their exit. When particles hang around like this, the neuron stops firing. As for the chemical signal, if too much chemical signal sticks around the next neuron, it can get overstimulated and die. Too much potassium and the cell doesn't fire. Too much signal and the neuron dies. In order for neurons to maintain their communication, these chemicals need to be cleared from the surrounding fluid. Without clearance, the damage to neurons can lead to dementia or epilepsy. A glial cell known as an astrocyte, or star-shaped cell, will support the neuron by removing potassium and signal from the brain's fluid. This allows the neuron to continue firing appropriately. This is one of the many ways astrocytes keep neurons healthy and monitor their connections. Other glia, called oligodendrocytes, wrap around neural wires to allow faster electrical communication. Oligo means many, and dendro refers to the branches of trees. Oligodendrocytes are literally many-branched cells, since their many branching arms insulate the wires of many neurons. So-called white matter in the brain is white due to the fatty insulation from these oligodendrocytes. Without oligodendrocytes, the electrical signaling of the brain would occur too slowly, and loss of these cells can lead to neurological diseases like multiple sclerosis. Finally, we have microglia, small cells that protect the brain. Microglia is a bit of a misnomer, as microglia are immune cells, more similar to white blood cells than other glia. Microglia will swim throughout the brain on patrol for anything that shouldn't be in there. If it encounters an invader, like a germ, it will activate and attack. Not only do these cells protect neurons, there's evidence that they may also regulate the connections between them, which, as mentioned before, are the basis of learning and memory. So neurons are cells that fire electrical and chemical signals and are responsible for memory, learning, and intelligence. Astrocytes keep neurons running by cleaning up after them. Oligodendrocytes allow fast communication between neurons. Microglia keep cells in the brain safe from diseases and regulate their connections. These glia are a crucial support system for the brain and may be important for intelligence. Now, why would that be the case? Well, since glia regulate neurons, it makes sense that they would influence the ability to store information in the brain. Other mammals, like pigs, mice, and monkeys, all have the same brain cell types as us. They also have neurons, astrocytes, oligodendrocytes, and microglia rattling around in their heads. Humans, famously, have larger, more complex brains relative to other animals, despite the similarities of our brain cells. Interestingly, while variation of neurons between species is relatively low, astrocytes vary greatly in size and shape between species. Humans, for instance, have much larger astrocytes with more branches than the astrocytes of mice. So why would we even want to put human cells in the brains of monkeys or mice? Well, one of the major goals of biomedical research is to better understand disease so we can develop treatments for those diseases. To do so, we often need to tinker with the organs damaged by the disease. Our institutions have deemed it unethical, usually, to give humans disease for research purposes, so we typically use other mammals, like mice. On autopsy, people with Alzheimer's have clumps of protein among their neurons. Research into curing this disease often focuses on targeting the proteins that make up these large clumps and understanding the role they may play in killing neurons. Microglia, the resident immune cells of the brain, are known to activate and clear these protein clumps, suggesting that they may have some ability to protect the brain from dementia. Studying how these cells work in the human brain may prove useful in fighting disease. Mice have microglia too, and we can grow human microglia in a dish, but in both cases, the microglia simply don't behave the same way as they do in the human brain. To study Alzheimer's, a team at UC Irvine grew chimeric mice with human microglia. They found that the human microglia in the mice behaved and looked like they would in the human brain. Here's a cross-section of the mouse brain with the human microglia labeled in green. The human microglia attacked protein clumps in the mouse brain, just as they would in the human brain. One could say that human cells in the mouse brain seem to function as they would in humans. That being said, the research did not look into any other human characteristics in the mice beyond modeling Alzheimer's. The human microglia also did not totally replace the mice microglia, though this is not the only time human cells were put into the brain of a mouse. A lab at the University of Rochester created mice with human oligodendrocytes, the insulating cells that speed up communication in the brain. In mice with incompetent oligodendrocytes, human cells entirely replace the mouse's insulation. In this image, we actually see the human cells, labeled here in green, gradually replace the red labeled mouse oligodendrocytes as the animal ages. In the chimeras we have described thus far, the human cells coexisted with the resident mouse cells, but these mice were bred to have busted oligodendrocytes. They were unable to adequately insulate neurons on their own. The human cell graft was then able to outcompete the deficient mouse cells. Here we have a mouse where a subset of its cells, a subset of its brain cells, are entirely human. Of course, when we think of brain cells, we think of neurons. And yes, scientists have put human neurons into the brains of mice. A study found that despite taking longer to grow, human neurons that were introduced to a mouse brain were able to wire together with the mouse neurons. The integrated human neurons seemed to even take part in processing visual information in the mouse. Humans live quite a bit longer than mice, so the human neurons did not fully mature into adult neurons. Still, the juvenile human neurons were able to connect with and respond to signals from the native mouse neurons, and were even able to help the mice see. This is a video of human neurons firing in a mouse retina. These Belgian scientists demonstrated that human cells can integrate into the neural networks of mice. We want to know if human chimera mice gain any abilities that we view as distinctly human. Homo sapiens, the name we humans gave ourselves, literally means wise man. Do human brain cells make mice more wise? The same team that created the human oligodendrocyte mice also created mice chimeras with human astrocytes. Recall that astrocytes were the cells that cleaned up after neurons and that are known to be quite more complex in humans than in rodents. Mice with human astrocytes had 4 times better memory and are faster at navigating mazes than normal-brained mice. The human cells in the mice were much larger and branchier than the native mice astrocytes. Here you can see a green human astrocyte among native red labeled mouse astrocytes. Human astrocyte mice were better able to form connections in the brain, better able to solve mazes, better able to remember locations of objects, and better able to associate a tone with an electric shock. Adding human astrocytes to mice makes them better learners. Stanford's Irving Weissman proposed making a human mouse chimera where all of the neurons in the mouse were human. Remember that in the previously mentioned human neuron mouse study, only a small subset of neurons were human. The glia would be mouse, and the neurons would be human, in order to study human neurons in a living being without killing an actual human for study. Weissman, a renowned professor of pathology, is known in part for creating a mouse with an entirely human immune system, allowing scientists to study diseases like HIV in mice. A mouse with an entirely human nervous system could be used to study neurological or psychiatric diseases like Alzheimer's or depression. Ultimately, the totally human neuron mouse was never created, but it does raise an interesting question. If a mouse with only a subset of human astrocytes is more intelligent, imagine a mouse with an entirely human-derived brain. So when human microglia are put into a mouse, the microglia act as they do in humans. Put human oligodendrocytes into mice with busted oligodendrocytes, and the human cells replace the mouse cells. Give mice human neurons, and those neurons will integrate into mouse neural networks and take part in facilitating sight and learning. Introduce human astrocytes into a mouse, and the mice become smarter. Put human brain cells into a mouse, and not only will the mouse survive, but it will develop with some, albeit limited, human characteristics. We've looked at human-animal chimeras. Now let's take a look at genetically modified animals. So far, we've talked about chimeras, animals with a subset of cells that are human. In the chimera, the animal cells have animal DNA with animal genes, and the small populations of human cells have human DNA with human genes. Typically, almost every cell in a normal animal would have the same DNA. Genetic modification allows us to engineer animals where every cell has some human DNA. The vast majority of each cell in the animal has animal DNA, but each cell may also have a segment of human DNA that we introduced. In humans, some of our DNA is involved in what makes us distinct from other animals. Some of that DNA is in genes that make us intelligent, emotional, or conscious, genes that make us human. What if we put that DNA into an animal, say a mouse or a monkey? To study how human brains are unique, scientists at Duke University created mouse embryos with a portion of human DNA and another line of mouse embryos with an equivalent portion of chimpanzee DNA. They found that the human DNA mice had brain cells that divide quickly, and created mice with 12% larger brains. Here is an actual image from the paper showing a human DNA mouse fetus with an exposed and enlarged brain, stained in blue. One of the major differences between the neurons of chimps and the neurons of humans is that during development, human neurons seem to divide more often. Another study, this time conducted by the Max Planck Institute, introduced a human gene to the smoothed brain marmoset monkey, and their fetal brains grew larger and more folded, like that of human brains. As you can see from this image, the human gene made the fetal monkey brain on the right wrinkle just a little bit. Once again, the researchers deemed it unethical to carry the human gene monkeys to term, so they were never born. In 2019, Chinese scientists did successfully create 11 human gene monkeys and allowed those monkeys to be born. These monkeys were given the human version of a gene that made their brains develop at a slower, more human pace. As mentioned earlier, human brains take longer to cook than the brains of other animals, and the presence of this gene made the monkey's brain cells take their time before maturing. The gene in question is called microcephalin. It is called microcephalin because people with a dysfunctional mutation in this gene are born with microcephaly. micro meaning small, and cephaly referring to the head, a disease which causes the brain to be underdeveloped, resulting in a small head. The microcephalin gene in humans is different from the microcephalin in monkeys, so scientists wanted to see if the human version of the gene, which seems to be implicated in brain size, would affect the intelligence of these simians. They found that monkeys with the human gene were, in fact, better at short-term memory. They tested the monkeys with a pattern recognition task to see if they could accurately remember the visual patterns, and it turns out the monkeys with the human gene had faster reaction times and better scores on the test than normal monkeys. Interestingly, the brain size of the monkeys were normal. The human gene did not make the brains bigger. Dr. Bing Su, the study's lead researcher, says the goal of the experiment was to understand the evolution of human cognition. He thought that putting human brain genes in monkeys might help us understand why humans are so smart. In an interview with MIT Technology Review, Dr. Su concedes that these experiments shouldn't be done on apes, but since monkeys are more distant, the experiments are ethical. Quote, "Although their genome is close to ours, there are also tens of millions of differences." Martin Steiner, a scientist from the University of North Carolina and a collaborator on the experiment, believes the study did not reveal much information and that it was unethical. He even considered removing his name from the paper. Steiner is quoted saying, "I don't think that this is a good direction. Now we have created this animal which is different than what it is supposed to be. When we do experiments, we have to have a good understanding of what we are trying to learn to help society, and that is not the case here. They are trying to understand brain development, and I don't think they are getting there." The role the microcephalin gene plays in intelligence is controversial, and although a handful of monkeys with the human gene seem to be decent at remembering a few colors and shapes, it isn't clear if this makes them more human in any meaningful way. It is important to emphasize that intelligence is a sort of ill-defined trait, and there isn't any one single gene that is the smoking gun. Human cognitive ability likely involves many genes working together, each playing a subtle role in the structure of the brain. What can we say about human-animal chimeras and genetically modified animals with human genes? Human stem cells introduced to the early embryo of a monkey seem to retain some human characteristics, but ultimately start to resemble the monkey cells that surround them. Introduce human brain cells to the brain of a more developed mouse embryo, and they seem to retain human functions. Mice and monkeys with human DNA involved in brain development have brains that develop like human brains. From these studies, we know that human cells and genes can exist in monkeys and mice. In the purported interest of solving the organ donation shortage, curing neurological disease, and understanding human evolution, scientists have created animals with human brain cells and pieces of human DNA. In some cases, it seems this improves the cognitive abilities of the animal. In other cases, the intellectual and emotional effects are unknown. Does making an animal slightly smarter make it more human? For some, the important question is whether these animals should be treated like humans. Do they suffer more because of their humanization? Do they understand that they are different? Do they feel like an animal life is lacking? These questions are difficult to study. Some are worried that we are creating a planet of the apes, while others believe that there is no precedent to believe that this is the case from the research done on chimeric and transgenic animals so far. Bioethicists emphasize the importance of a well-informed public when it comes to questions of chimera research and genetic modification. And to have an informed opinion, one must understand the methods and results of said research. One can find this information by clicking through the podcasts and pop science articles to the actual primary source, the scientific articles that detail the experimental procedures and discuss their findings. Thanks for watching, and as always, sources are in the description. Take care and goodbye. Bye-bye. Bye. Kisses, darling. Goodbye. Yes.